I came here to discuss about this drugs used in COVID-19 pandemic. Myself, Dr. Manohar, professor in VGS College of Pharmacy. Coronavirus case worldwide is 23,32,450. If you see this, the recovered patient is 25.72% and deaths around 6.89% as up to April 19th. So here I would like to explain you different drugs used by different countries and their mechanism of action. So before going to this, just the introduction. The COVID-19, it is a RNA virus. It requires host machinery to prepare its virions. Normally it uses our ribosomes and various proteosomes to prepare virions. Virus can spread through the biological fluid, unhygienic behavior, fecal contamination, etc. It uses the spike proteins to enter the cells. Here first we'll discuss about the pathophysiology of the coronavirus. So this is very important because to target any particular protein, it is mandatory to know how the virus is infecting our host cell. So first we'll start with pathophysiology. So the virus first enters through these ACE2 receptors, angiotensin converting receptors, and it will form the endosomes. The blue color appears is the endosomes. So lysosomes are going to be attracted towards this endosome, and it releases its contents, where these contents can metabolize the lipid membrane around this virus, and the releasing of the nascent RNA. So this RNA is first translated by using host ribosomes to RNA-dependent RNA polymerase. So the function of this RNA-dependent RNA polymerase is to make multiple copies of the RNA, and these RNAs are going to be translated and form various structural and non-structural proteins by using the Golgi operators. These are going to be assembled and the new virion is going to be synthesized. Some of the proteins can translocate into the nucleus, and it will regulate the antiviral protein synthesis. Thereby it will inhibit the immunity activity against this virus. So here I would like to explain you different drugs used by different countries and their mechanism of action. So first we'll discuss about the interferon alpha. Interferon alpha consisting of around 165 amino acids. These are antiviral protein. It binds with the interferon receptors, which dimerages activation of the genase kinase and tyrosine kinases. Further, these are going to be activate STAT1, STAT2, which dimerages and is going to be translocated into the nucleus and activate various antiviral proteins. So this is the schematic representation. So when interferon is bind to its receptor, this JAK-STAT pathway is going to be activated. The STAT is going to be form complex with ISGF complex, which translocated and is going to be initiate activity of interferon, where the antiviral proteins is going to be synthesized. So coming to this Lopinavir and Ritonavir. So these drugs are given combinedly. Lopinavir is an antiretroviral protease inhibitors. It is uses against normally it is anti-HIV drugs. Ritonavir is used to inhibit the cytochrome P450-34A in intestine, liver, which metabolize the protease inhibitors. So this is the basic structure of Lopinavir and Ritonavir. So normally here it will inhibit the host proteolytic enzymes. So these enzymes are very essential for the breakdown of the the proteins of the virus to structural non-structural proteins. When it is not cleaved properly, then there won't be a proper assembly of the virus. So resulting of that, the virus is going to be. So here protease inhibitors, the specific cytochrome P450 enzymes 3A4 is going to be metabolized for antiviral proteinase inhibitors. So, in presence of Ritonavir, so the Ritonavir is going to be bind with specific cytochrome CYP3A. So resulting of that, the metabolism of the Lopinavir is going to be decreased. So resulting, the activity of the Lopinavir is going to be increased. The drug is claimed to inhibit the viral entry into the target cells and stimulate the immune response. The drug stimulates a humoral immune response, induces the interferon production, and stimulates the phagocytic functions of the macrophages. So this is the schematic representation where the Erythromycin is going to be inhibit the entry of the virus. So thereby it will prevent the Ribavirin. Ribavirin is a prodrug which, when metabolized, resembles the purine RNA nucleotide. It is a guanosine analog used to stop the viral RNA synthesis and viral mRNA. It interferes with the RNA metabolism required for the viral replication. Ribavirin is basically decreases the nucleotide pools, so resulting of that viral replication is going to be decreased, and it causes the mutagenesis in mRNA, so thereby the virus is going to be died. And other mechanism is going to be increase the activity of the interferon. So that is the reason normally we'll use interferon and ribavirin for the treatment of hepatitis C virus. So here in ribavirin initiate the activity of interferon. At the same time, the viral replication is going to be inhibited because of the mutagenesis and decreased nucleotide pools. Remdesivir. Remdesivir is an adenosine analog, inserts into the viral RNA chain, causing their premature termination. It interferes with the actions of the viral RNA-dependent RNA polymerase and evades the proofreading by the viral exoribonucleases, causing a decrease in viral RNA productions. So here there is a similar structure of ATP and remdesivir. So resulting of that, it shows activity. So here remdesivir is specifically bind RDRP, RNA-dependent RNA polymerase, resulting the multiple copies of the RNA is not going to be synthesized and is going to be inhibit the RNA synthesis. Favipiravir is an antiviral drug that selectively inhibits RNA-dependent RNA polymerase of the influenza virus. It is phosphoribosylated by the cellular enzymes to its active form, favipiravir ribofurosyl 5-triphosphate. So which resembling the nucleotides. So resulting of that is it inhibit the viral replication. In various countries, and various clinical trials are going on which act on this COVID. But no drug is not approved, and here I tried to make you explain the mechanism of action of various drugs. Hope you understand. Now chloroquine phosphate. So there is a tremendous need worldwide regarding this chloroquine. Chloroquine increases the endosomal pH and interferes with the glycolization of the cellular receptors of SARS-CoV. Chloroquine also inhibits the quinoline residues too, which involves in the sialic acid biosynthesis. So this is the diagram. So normally this chloroquine is a highly basic drug, so it's going to be it's deposited in endosomes and lysosomes. So when the lysosomes are bind with this endosomes, so the outer layer of the virus has to be digested properly, but in the presence of chloroquine, so the digestion is not appropriate, resulting of that, because the chloroquine is going to be increase pH, so resulting the outer layer digestion is going to be decreased, so nascent RNA cannot be released. That is one of the mechanism. And second mechanism is chloroquine is enhance the entry of the zinc ions into this cytoplasm. So when the zinc ions, the yellow particles which is appeared on the screen, is enter into the cytoplasm, is bind to RNA dependent RNA polymerase. So because of this inhibition of RDRP, so there won't be any replication of the RNA. So thus the chloroquine can inhibit the replication of the virus. So normal the mechanism of action of the chloroquine is it is an antimalarial drug, so it will inhibit uh, the polymerization of the heme, so resulting of that the heme can kill the parasite. Infection. Ivermectin. Ivermectin is an anti-helminthic, act by the increasing the permeability of the parasite cells. It increases the chlorine influx into the parasites. Coming to this antiviral property, ivermectin is an inhibitor of interaction between the virus integrase protein and importin, heterodimer responsible for the nucleus import. A single treatment able to affect 5000 fold reduction in virus at 48 hours in cell culture. So I mean to say this is a promising drug which decreases the virus in a drastic way. So coming to mechanism of action of this ivermectin. So normally what happens when the viral proteins are synthesized, structural and non-structural proteins, some of the viral particles are combined with importin A and B and form a complex. So when this form a complex, these are going to be translocated into the nucleus and is going to be inhibit antiviral proteins. So resulting of that, the antiviral protein synthesis is going to be decreased and viral replication is going to be enhanced. But in case of the ivermectin, is going to be bind with importin A and B, so that the viral protein cannot bind to these particular translocate proteins. So resulting of that, the antiviral proteins are going to be increased. That results the degradation of the virus. Thank you. Stay home, stay safe.